All right, are there any questions? So today we are going to talk about expression microarrays, right? And the key feature of this is that you're going to measure the activity of thousands of genes at the same time, okay? Until about 15, 16 years ago, that was not possible. So biologists had to study the activity of one gene at a time, okay? But this microarray technology allows you to study the activity of thousands of genes at the same time, okay? So now, see, you have three levels in the central dogma, right? Like you have DNA, right? Then you have RNA, right? And then you have protein, okay? There is the interaction between all the three levels, okay? So, there will be information available in each of these levels, and they will also be mixed up because proteins interact with DNA and RNA and so on, okay? But usually, as far as microarrays are concerned, right, you focus at the RNA level, right? So you're looking at the messenger RNA that is being expressed in a particular tissue, right? And based on that, you're going to try to figure out the activity, right? Now, one thing to note when we are, and why do you want to study microarrays? Because usually cellular control results from multivariate activity among cohorts of genes and their products, okay? The biologists normally look at it as one gene, right, affecting another one, okay? But that's not the story. You have lots of genes that are interacting with each other. So you want to get the overall picture, right? And for that you need something like a microarray, right? Now since all the three levels in the central dogma, DNA, RNA and protein, they interact, it is not possible to fully separate them. And information, in fact, from all these domains, like the DNA, then messenger RNA and proteins, must be combined for a full understanding, right? But the high level of interactivity between these levels also ensures that a significant amount of the system information is available in each of these levels. So that focus studies on a particular level, right? If it is at the RNA level, right, it'll be called transcriptomics, right, which is what you would do with microarrays. If it's at the protein level, if you're looking at the activity level of different proteins, right, that is proteomics, right? You may have heard of that, right? All these omic technologies, right? Genomics, transcriptomics, right? Genomics would be about at the DNA level, right? The genes, right? RNA level, RNA, you have the transcriptome, right? That is basically the set of all the RNAs that are present in a particular in a cell at a particular time. That's the transcriptome. So the study of that is transcriptomics, right? And if you're looking at all the proteins that are there, that is proteomics, right? And you want to take it a step further, many of these proteins are metabolites, right? So you'll have something called metabolomics, right? So there are people, really you're interested in the metabolomics, right? But, you can look at any of these levels, right? Now, most of the current effort is focused at the RNA level owing to measurement considerations, right? Now, again, see, you have to be able to measure, if you're going to look at the protein, right? If you're going to look at the activity of proteins, right, in proteomics, then, in order to go looking for a particular protein, you will have to figure out its antibody, right? Remember, antibodies were the molecules that would bind proteins with great specificity. So you have to, now for all the proteins, antibodies are not developed, right? But for RNAs, if you're going looking for RNAs, right, you can take the complementary RNA sequence and use that as a probe. Okay, that's the main idea, right? So if all the proteins had antibodies developed, then probably you would go to proteomics and work with it. And there are people who work on that, right? So high throughput technologies make it possible to simultaneously measure the RNA abundances or the RNA activity of tens of thousands of messenger RNAs at the same time. Okay, that's the power of the microarray, and in the media and all that, it is popularly referred to as the gene chip. Okay, just like in integrated circuits, you have a VLSI chip here. This is called a gene chip. Right? Now, again, let me go to a picture first, then I will go through the text, right? Because so this is a microarray flowchart, right? And this is a picture that is taken from an old paper, a 1997 paper by Mike Bittner, Yidong Chen, Mike Bittner, and Ed Doherty, right? So in this paper, and in fact, this is the first paper that analyzed microarray images, right? Did the image analysis for microarrays, right? So what is the idea here? And using this picture, it will be very easy to explain. See, first of all, you are going to go and look for certain genes, right? And you're going to see whether certain genes are expressed, right, in particular tissue, right? So the starting point is, so that means you need the complement, right? You need the complement. So in order to figure out whether a gene is expressed in a tissue or not, you will basically look at the messenger RNA, right? Because if the gene is turned on, it's expressed, then that tissue is going to produce the messenger RNA. Okay, so so you're going to look at the messenger RNA. Now, this is the glass slide that you're going to use, right? So you can see that there are thousands of spots on this glass slide. At each location, right, you have immobilized the complement of the gene that you're interested in, right? The gene whose expression you're interested in. So how would you prepare this glass slide, right? You have two options, okay? You can either put synthetic DNA over there, right, using what are called oligonucleotides, right? Or you could go ahead and, if you're interested in a particular gene, get the complement of that gene, right, the complementary DNA sequence, and immobilize it on this glass slide, all right, at different locations. So, so first, the preparation of the microarray would go as follows. If you're going to look at messenger RNA activity, all right, then you will go ahead and take the DNA, right? The genes that you're going to put here, right? You're going to amplify them. Maybe they are in small amounts. You use PCR to amplify, and then you will immobilize them. You will basically put them on different spots here, okay, using some robot or some device, and those devices are available. And again, this technology is also 15 years old. Okay, so right now there are other things people use too. So that's how you would prepare the microarray. Now, let's say you're going to compare two different specimens. Okay, one is coming from infected cells, the other one is from uninfected cells. Okay, now you want to see, is a particular gene expressed in the infected versus the uninfected tissue. All right, so you will collect messenger RNA from that tissue. All right, now messenger RNA is a very, very unstable molecule. Right, so you will convert it into complementary DNA. Right, how? By doing reverse transcription. Right, so there is this enzyme called reverse transcriptase. Right, that will convert this messenger RNA into complementary DNA. Okay, DNA that is complementary to it. While you're doing the conversion, you will make sure that the two samples are labeled differently. The complementary DNA that is coming from the infected cell, it has got some color tagged onto it. The one coming from the uninfected cell, it has some other color tagged onto it. Usually red and blue, red and green. Okay, so that way now you have this complementary DNA, right, with color tagged on or fluors tagged onto it, right? One is green, one is red. Now you take this stuff and apply it to each and every spot from each of these samples, okay? Both infected cells, uninfected cells. Take this material, apply it to each and every spot, okay? Where there is hybridization, right, they'll form the double helix. Where there is no hybridization, it will be just the single-stranded molecules. In the next step, you wash it away, right? You wash it away from here, right? So whatever was single-stranded, that gets washed away, right? What remains, because the DNA double helix is a very stable molecule, right? And that's the one that is going to stay. And of course, you can guess that with this amount of washing, mixing, and all these things, all right, there is going to be uncertainties that are going to be introduced, okay? Errors that are going to be introduced, and you need to properly account for them. So anyway, after this, you wash away, all the cDNA molecules that have failed to hybridize. And now, see, if at a particular location, let's say the infected cell is labeled, let's say, with red, the uninfected with green, right? So and let's say if a particular gene is expressed at a high level, right, in the infected tissue, right, then at that spot you will be having more of those red complementary DNAs hybridized, okay, and less green, right? So that's true for each and every spot, right? If the two are expressed at about the same level, then you'll roughly have the same amount of the red and green getting hybridized, okay? And of course, here the assumption is that the DNA that you have in here, right, is actually in excess. That means at each spot you have enough DNA, complementary DNA, to capture whatever samples you apply to it, right? You're making that. So those are all assumptions. They may or may not hold, right? So once you have hybridized, so at each location you have red and green molecules sticking onto it. Now you can do excitation with green light, with red light using lasers, right? And then you can create these two images, okay? These two images are telling you, if at a particular spot I see green is more intense than red, okay? Then I know that that particular gene is expressed at a higher level, right? In, let's say, the uninfected tissue versus the infected tissue, right? And so then with these images, you'll have to do some kind of image analysis, because this stuff, I'm talking about one green being higher than the red, you can see with the naked eye, right? But in reality, right, there may be cases where it will be a close call, okay? So you need some mathematical procedure, right? So there will have to be some kind of hypothesis testing, right? So the key question is red higher than green, right? Or it's about the same as green, right? Or is red smaller than green? So you'll need some hypothesis testing with some confidence interval, right? And for that you have to calculate some density functions, right? If you, right, from your probability statistics you know that, okay? You'll have to calculate some density function so that you can come up with a confidence interval, right, for this hypothesis test.
So what's the purpose of the experiment?
No, the purpose of the experiment is to find out the differential expression, right?
Oh,
Like, yeah, you want to find out, you have two different samples, let's say cancer versus normal tissue, okay? You want to find out, right? You don't want to focus on a particular gene. Across all the genes, okay, of interest, okay, which are the genes which are differentially expressed between these, okay? Cancer versus normal, okay? And to find that out, if they are differentially expressed, then the red and the green intensities will be significantly different, okay? That's what you're trying to determine, okay? So, this paper does that, okay? It comes up with the density function and it'll make use of the density function to come up with confidence intervals, right? And what is your goal? Once you find out at every location, right? So this is going into the engineering issues because at every location, let's say 1 means, the red is significantly higher than the green. 0 means red and green are about the same, okay, within that confidence interval, right? And let's say -1 means that the red is significantly smaller than the green, right? So red significantly smaller than the green, red is about the same as green, right? Red is much higher than green, right? So at each spot, all right, you will put in a number, all right, 1, 0, -1. So you're basically classifying the response at each spot, okay? Now, based on this, suppose you have trained a classifier, right? You gave it a lot of samples like this, cancer versus normal, okay? You want the classifier to get trained so that when you give it a new sample, a new microarray, it can correctly say whether it is cancerous tissue or normal, okay? So believe me, because if you show the same slides to the pathologist, okay, today and 6 months from now, they'll come up with a different answer, okay, because the human bias is there, okay? But if you have a classifier doing the job, it's going to be automated, okay? Now, if you're going to do classification, all right, if you have 10,000 genes on this, okay, are you going to use all the 10,000 genes as a feature vector? No. So you have to choose some features, okay? Which is the small set of genes that you're going to focus on or features that you're going to focus on so that you can get good classification, right? Another problem is that if you're familiar with the theory of classification, right, I mean, as the number of samples becomes very large, right, you have a lot of asymptotic results, okay? Problem here is that nobody that has got cancer, okay, before getting cancer, people don't go to like MD Anderson or somewhere every month, hey, take some tissue from me, okay, create lots of samples, so that if somebody gets cancer, they can use that later. So your sample size is very small, okay? So all those asymptotic theorems do not work anymore, okay? So how are you going to do it in the case of small samples, okay? So that's another challenge, okay? That is there all throughout. So at least I hope you're liking it because you're now getting into double E problems, okay? These are not biological problems anymore, okay? Then also we want to find out, okay, we want to use this information, let's say this pattern of 1, zeros and -1s, all right? We want to use this information to figure out relationships between genes. This is not like a typical engineering system, okay, where I know the physics behind it, right? Let's say if it's a, um, mass viscous damper or a spring system, okay, I can write down the equations. I know the physics, right? Then I get the data. Here I just have some data. Question is, can I use this data to figure out, right, which genes are related? Well, if you look across many different conditions and you see some genes going up and down together, maybe there is some relationship between them. You don't know what it is, okay? Right? So let's say if you look at microarrays across different conditions, all right, and some genes never change, maybe those are not important, right? So you can use some techniques to come up with relationships between the different genes. And the idea is, I mean, after that, if you want to, if you're going to let it evolve as a dynamical system, right? You try to, model this as some kind of a dynamical system, like using maybe state equations or something like that, right? The behavior of the genes, right? And then maybe you can even do some control on that, okay? Let's say the pattern corresponding to cancer is bad, okay? You have a certain pattern. You want to get it to something else, right? So the question is, can you intervene with drugs or something else to, get it out from a bad state to a good state, right? Again, all this is easier said than done because this is a very challenging problem, right? Because you do not have a model for the system, right? For the cell, only parts of the signaling mechanism are understood. There are many things that are not understood, okay? So, and we don't understand all the basic physics and all that stuff, So it's like more like signal processing, all right? Looking at the signal, all right, I'm going to construct a model, all right? And hopefully that will have predictive power, all right? So the microarray is like the first step in this. The microarray technology is not very good in the sense that it has got all these different sources of possible errors. Okay, you're doing washing, you're mixing things together and all that. Okay. Uh, the other thing is that in order to get the messenger RNA, okay, typically what they do is they will go and take that tissue, all right, subject the cells to all those different procedures. Remember I talked about how you break up cells and then mix things up and all. They'll mix everything up really well. So you have killed a cell, right? So this is like an invasive thing. It's a one-time thing, right? And when biologists talk about time course data, what they mean is that like maybe they have 100 rats, okay? They will kill 10 of them at this point in time, another 10 later on, and they talk about that as time course. This is not from the same individual, right? But today so this is not capable of following a single cell, all right? It's not capable of providing time course data on a per cell basis, right? Here at the center, we do have the technology right now, right, to generate that kind of data. Like Mike Bittner from TGen, right? He's moved his lab here. He can do that. Okay, how does he do it? Remember, we talked about the promoter region of a gene, right? Does that ring a bell? Promoter region of a gene, right? So the RNA polymerase has to tag onto the promoter, okay, in order for the gene to turn on, right? What he has done is, he has basically put, so there is something called a green fluorescent protein, right? That is usually extracted from jellyfish, right? So he has put in the gene that codes for the green fluorescent protein under the control of the same promoter. Okay, so every time this gene turns on, okay, the promoter becomes active. The green fluorescent protein also is transcribed and translated. Okay, so he's able to capture that green light and see what's going on. So initially, of course, he has to tag the cell with the green fluorescent protein, the promoter region. But then after that, he doesn't have to kill the cell. Okay, he can get that data. Okay, and you can follow it over time. Okay, so that technology is there. I mean, like some of you might be interested in working on that, and I'm just telling you, it's a pretty good deal that 6 weeks into the semester I'm able to tell you what is available here. Okay, that you can go and connect to that stuff. If you're interested. Right? But we'll go through this microarray chapter. So what I have here is a general description of all these steps, okay, in this chapter, and then I will discuss this paper also in some detail because all of you are grad students, all right? So this paper I will probably discuss next time, okay? How to derive the equations, all right? You will see maximum likelihood estimation, all right? Then all these rank sum tests and things like that, okay? Hypothesis testing, confidence intervals and all. So things that are kind of closer to your comfort zone, right? All right, any questions? So then now that I've shown you the picture, I think whatever is written before is going to make sense as we walk through it. Right? All right, so expression microarrays result from a complex biochemical optical system incorporating robotic spotting because those spots, okay, in the microarray preparation, those spots, usually a robot will do it, okay. Then again, I mean, if there's imprecision with regard to the location of the robot, pen, right, there'll be some, errors that could be introduced. So these arrays, so complex incorporating robotic spotting and computer image formation. I already explained to you how that is done. So these arrays are grids of thousands of different single-stranded DNA molecules attached to a surface to serve as probes. Right? That's the complementary DNA. Two major kinds are those using synthesized oligonucleotides and those using spotted cDNAs. Right? So now let me just point out, something else also. This was not there in the initial paper. So here, what I'm talking about is with complementary DNAs. Okay, so the things that you'll be putting in here, that is basically a gene. That is a complement of some gene. Okay. On the other hand, somebody might say a gene is too long. Okay, I'm going to put pieces on there that are only 25 nucleotides long. Right. But so in that case, a single piece here is not going to cover an entire gene. Okay. One gene will span across many of these complementary pieces because each one is 25 pieces. But I have to have some algorithm, right, that will collect that information and then give me the information about the gene, right? So there are companies that have done that, like one example being Affymetrix and others, all right? They have like these short stretches, all right, that will hybridize, all right? And based on that, and there might be overlap also between one gene and the other, but they have their own proprietary algorithms that they will not give to you, all right? Like they'll give you a formula, they will sell this chip, and then they'll tell you take your sample, hybridize it. Okay. Now look at the intensity at each of the spots. Okay. Multiply the intensity at spot 5 by zero point 0.1 and then by some other number, all right, at some other spot, and then you just get your answer. Okay. That's how this technology is done today. Right. Okay, so two major kinds are those using synthesized oligonucleotides. Oligonucleotides are synthetic DNA, basically, right? And those using spotted complementary DNA. So they'll come from the messenger RNA from particular tissue. So if you're doing the one with cDNAs, all right, so you'll hear the terminology cDNA microarrays. That means complementary DNA microarrays. And about 10 years ago or so, it was the thing. Okay, I mean it was very hot. Okay, everybody was doing microarrays at that time. So the basic procedure is to extract RNA from cells, convert the RNA to single-stranded complementary DNA using reverse transcription, attach fluorescent labels, I showed that to you on the picture, to the different cDNAs, allow the single-stranded cDNAs to hybridize to their complementary probes on the microarray, and then to detect the resulting fluor-tagged hybrids via excitation of the attached fluors and image formation using a scanning confocal microscope. So relative RNA abundance is measured via measurement of signal intensity from the attached fluors. Okay, and that's where the double E's come in. Okay, you're going to do image analysis, do some measurements. So the intensity is obtained by image processing and by statistical analysis. Particular attention is paid to the detection of high or low expressing genes, and beyond that to expression-based phenotype classification. I talked about that. It was 1, 0, minus 1. Okay, you're trying to classify cancer versus no cancer. Okay, and to the discovery of multivariate intergene predictive relationships. All right. I also talked about, these are things that we'll cover in detail. Okay, some techniques have been developed, and there are later chapters that are devoted to that. All right. So in this chapter, we briefly discuss microarrays, their hybridization-based foundation, right? Then normalization, and also ratio analysis, right? Okay, hybridization-based foundation, because that's the basic property, right? The DNA is going to hybridize to its complementary sequence, right? The strand, DNA strand. Then you might need normalization because, see, you might have a red channel and a green channel, right? I mean, two different. Now, the way you've prepared, okay, the two different samples, okay, there might be variation over there. On top of that, even the laser that you're using, maybe the red laser is twice as powerful as the green laser, right? In that case, you know, if you see an intensity in the red laser that is 2, that should correspond to an intensity in the red laser that is 1. So the ratio that you would be looking for will not be 1 in that case, it will be 2, right? If you deviate from 2, then that's the problem. Okay, so that's the normalization. And then ratio analysis, because the ratio is the crucial thing. Ratio of red to green intensity. Okay, that is going to decide whether the particular gene in question is differentially expressed or not between the two samples. So the principle behind cDNA or complementary DNA spotted arrays is that an mRNA transcript to be detected is copied into a complementary DNA, cDNA, and this copied form of the transcript is immobilized on a glass microscope slide. Right? And the slides are usually coated with polylysine or polyamine to immobilize the DNA molecules on the surface. Right? So you basically have a glass slide, it might be 1 inch by 1 inch, it's got thousands of spots on it. Right? So for robotic spotting, the robot is going to dip its pins into solutions that contain the cDNA, and then the tiny amounts of solution that adhere to the pins are transferred to the surface. Right? And each pin produces a printed spot containing cDNA. Another method uses inkjet printing, in which the cDNA is expelled from a small nozzle equipped with a piezoelectric fitting by applying electric current. Right? And again, I mean, these technologies are like more than 10 years old. Okay? Today maybe they're doing something else. Right? The company will just give you a chip and you'll work with that. So a complete cDNA microarray is prepared by printing thousands of cDNAs in an array format on the glass slide, and these provide gene-specific hybridization targets. Right? And a schematic representation of the preparation, hybridization, image acquisition, and analysis for cDNA microarrays is shown in the figure. That's the figure that I showed you a few minutes ago. Right? Now, a digital image reflecting the abundance of different mRNAs in a sample is formed in a number of steps. Right? So in the beginning, it was just a microarray preparation. So that was the picture to the left, right? The leftmost picture. So now you got all these complements immobilized on the surface. Now you're going to prepare the samples, all right, that you're going to study, all right, and then get them to hybridize on that slide. Right? So the first step in getting that digital image is that RNA is extracted from the cells of interest. It is converted to complementary DNA by reverse transcription, and then it is amplified, right? Because maybe the amount of DNA you got is very small, okay? So you want to amplify that. So now PCR comes into the picture, polymerase chain reaction, right? So by a reverse transcriptase polymerase chain reaction. And over here also, you are interested in messenger RNA, okay? What is the unique defining property of eukaryotic messenger RNA? Does anybody remember? Poly A tail. So by focusing only on the poly A tail when you're doing the reverse transcription, right? You are going to make sure that you pick up only messenger RNA and nothing else, okay? Right? So during the process, fluorescent molecules are also attached to the DNA, to the new DNA that is being formed. So if a specific mRNA molecule is produced by the cells, then the generated fluorescently labeled cDNA molecule will hybridize to its complementary single-stranded microarray probe. Right? And now you would be in a position to shine light on it and see how much of hybridization has taken place. The cDNA molecules that do not find their complementary single-stranded DNA sequences on the microarray, they are removed in a washing step. Since the fluorescent tags are attached to the cDNA strands that hybridize, the corresponding spots will fluoresce when provided fluorescence excitation energy and will be detected at the level of emitted light. Right. And this yields a digital image whose intensities reflect levels of measurement fluorescence, which in turn reflect mRNA abundances. Okay, how much of that mRNA was there in the original sample, right? So in practice, it is commonplace to label mRNA molecules from distinct sources with different colors, and then to co-hybridize them onto each array gene. And two monochrome images are obtained from the red and the green are obtained from laser excitations at two different wavelengths. So monochrome images of the intensity for each fluorescent label are combined by placing each image in the appropriate color channel of an RGB image. Okay, so those of you that are image processing people here, you'll know what is involved here. I'm not an image processing person. Okay, you are.
I use RGB.
You learned some of it, yeah, yeah. What RGB is what red, green, blue, right? Yeah. Okay. So in this composite image, one can visualize the differential expression of genes in the two cell types, the test sample typically being placed in the red channel with the reference sample in the green channel. Intense red all this is stuff that I already told you. Intense red fluorescence at a spot indicates a high level of expression of that gene in the test sample, okay, because the test sample has been labeled with red relative to the reference sample. Conversely, intense green fluorescence at a spot indicates relatively low expression of that gene in the test sample compared to the reference sample. Now, when both test and reference samples, they are expressing a gene at similar levels, the observed array spot is yellow, right? Now here there is a bunch of assumptions that are made before you can do the analysis. So assuming that specific DNA products from two samples have an equal probability of hybridizing, right, to the specific target, the fluorescent intensity measurement is a function of the amount of specific RNA available within each sample, provided that the samples have been well mixed, right? And there is sufficiently abundant cDNA deposited at each target location, right? So assuming that everything has been properly latched on, then you can come to this conclusion that you are measuring the RNA intensity. Ratios or direct intensity measurements of gene expression levels between the samples can be used to detect meaningfully different expression levels between the samples for a given gene. Now, there is image processing involved. So when using cDNA microarrays, you will have to extract the signal from the background, right? And this requires image processing to extract signals, variability analysis, and measurement quality assessment, right? All these things are not, foreign to us engineers, okay? You have to do all these things. So the objective of the microarray image analysis is to extract probe intensities or ratios at each cDNA target location, and then to cross-link printed clone information, right? So that biologists can easily interpret the outcomes and high-level analysis can be performed, right? So you get that information, then you pass it on to the biologists, so that they can say, oh yeah, this relationship looks like what I already knew, or this is something new, okay? Now a microarray image is first segmented into individual cDNA targets, either by manual interaction or an automated algorithm. So if you've done image processing, there are algorithms that are available for segmentation, right? So for each target, the surrounding background fluorescent intensity is estimated along with the exact target location, fluorescent intensity, and expression ratios. Right? All these things you have to calculate. So this is double E stuff, no biology. Right? As with any random data, understanding variation is important to microarray data analysis. A major impediment to an effective understanding of variation is the large number of sources of variance inherent in microarray measurements. Okay, and I gave you examples of that. You're washing away, you're mixing things, okay, getting it to hybridize. So every stage there is uncertainty. Now, in many statistical publications, analysis publications, the measured gene expression. So you can imagine, when this microarray technology came into being, this is fertile ground for the statisticians, all right? All the methods that they know, they can now apply to microarray data and get new papers. And there are people who have done that, okay? I don't think they left any papers for you and me to do. It's all done, all right? So in many statistical analysis publications, the measured gene expression data are assumed to have multiple noise sources, like noise due to sample preparation, labeling, hybridization, background fluorescence, different arrays, fluorescent dyes, and different printing locations. Maybe you put one more variation in there and write one paper, okay, so if there's something not covered. Now, various approaches have been taken to quantify and treat the noise levels in a set of experiments, including, and again, these are all the different models that are there in statistics, okay, log transform, signal plus noise. ANOVA is an analysis of variance models, okay, if you've done experimental design, you'd know what that is, okay. Mixture models, all right, multiplicative models, ratio distribution models, rank-based models, less sensitive to noise distributions, replicates using mixed models, and quantitative noise analysis, okay. So it's probably not a good idea now to think of getting a PhD writing that next paper on statistical analysis of, because those guys have published, but now if you try to put your paper in, they'll give you a hard time, okay, because they'll say that we did the work, you don't need to do anymore, okay. Now, besides variation due to random effects, such as biochemical and scanner noise, simultaneous measurement of mRNA expression levels via cDNA microarrays involves variation owing to system sources, including labeling bias, right? Imperfections due to spot extraction and cross hybridization, right? There could be other sources of error, like the photomultiplier tube. The laser might have a different intensity, right? So things like that also. Now, even with the development of good extraction, that is image extraction algorithms from the background, and the use of control probes at the array printing stage to aid in accounting for cross hybridization, we are left with labeling bias resulting from the fluorescent tags as systemic error, right? Because you're using two different tags, okay? Maybe they have different efficiency of binding, right? The red and the green. That will cause problems for you. So although different experimental designs target different profiling objectives, whether it be global cancer tissue profiling, right? That means you're trying to find out how the gene activity varies across cancer tissue, right? Or whether it's a single induction experiment with one gene perturb. Maybe you went and messed around with one gene and you want to see what other genes are getting, affected, right? Normalization to correct labeling bias is a common preliminary step. This is what I was talking about. If one of the lasers has higher intensity than the other, right, then you have to do some kind of normalization. Okay, you cannot just compare the red and the green data without doing the normalization. So normalization to correct labeling bias is a common preliminary step before further statistical or computational analysis is applied. And lots of papers have been written on this normalization of microarray data. So the objective of this preliminary step is to reduce the variation between arrays. Right? So normalization is usually implemented for an individual array and is then called intra-array normalization, which was what we will consider here. Assessment of the effectiveness of normalization has mainly been confined to the ability to detect differentially expressed genes. Okay, so if you normalize, you're in better shape. Okay, because if the red laser is more intense than the green laser, okay, you don't normalize, you're not going to get correct results. Okay. So it has also been shown that normalization can benefit classification. All right, remember I said that there's -1, 1, and zeros. They are going to be used to classify, all right, disease. All right. So if you do the normalization, it has been shown to also benefit classification accuracy, and the significance of this benefit depending on the bias properties. Right. So let's look at normalization of microarray data. The simplest and most commonly used normalization is what is called the offset method. All right. To describe it, let the red and green channel intensities of the K gene be denoted by R sub K and G sub K respectively. In many cases, these are background subtracted intensities. Now, in an ideal case, okay, if at the same spot, okay, you are just using the same biological samples, two identical biological samples are used. Same sample, you break it up into two parts, all right, red and green, and then you put it on the same spot, okay. So ideally, you should not see any difference, right? I mean, red, the intensity of the red should be intensity of the green at that location, right? So which means that the log of that should be 0, right? R over G, if that is 1, the log should be 0, right? If it's different from 0 means you need some correction, right? So in an ideal case where two identical biological samples are labeled and co-hybridized to the array, that means both the same sample, okay, or identical sample is put on the same spot. We expect the sum of the log transform ratios to be 0 because the log transform, before you do the log transformation, the ratio is 1. When you do the log transformation, it's 0. You sum it up, it should still give 0. However, due to various reasons such as dye efficiency, scanner PMT photomultiplier tube control, etc., this assumption may not be true, and we want to correct for that. So if we assume that the two channels are equivalent, except for a signal amplification factor, then the ratio of the kth gene, tk, can be calculated as follows. So you'll take this equation. See, the log of the ratio for the kth gene is basically log of the intensity of the red divided by the intensity of the green, right? But then you do a correction because even at those spots where the ratio was supposed to be 1 and the logs were supposed to sum up to 0, they didn't. Okay, so you go ahead and do that correction. So minus 1 over NQ, right? NQ is the number of probes that have measurement quality score of 1. Okay, that means red over green is 1, right? So at those probes you really got something else, all right? So you take this log of RK over GK, sum it up, divided by NQ, and do a correction term, right? So
Red over green means is 1, or body score of 1. Because if that's the case, then log 1 will be 0.
exactly. No. See, these are the spots where you should have 1, okay? But you will not get, you're not getting 1 because there's something wrong, right?
Right. So we just subtract the error.
Yes, error, yeah. All right, any other questions? So, in fact, if you look at the, I think there's a picture here, see? This is the offset, okay? See, there are effects of normalization. Left-hand scatter plot shows the regression lines, and the red scatter plot show the normalized scatter plots. So this is offset, okay? So if you don't do any correction, you're not going through the origin, okay, the red and green, okay? You do the correction, you're going through the origin. So this is offset, then there's, you might just do a shift, okay? Then in some other case, you might need to do a line. It's not just the y-intercept, okay? If the slopes might be different, so then you do a linear regression, right? And then another one, there is a variation of that is called low S, okay? And there are lots and lots of these, so there's no point in us going into details. For each of these, there will be several papers that people have written, because those were in the early days of microarrays, so people made a livelihood. Every new method, that they knew, every method that they knew, they basically tried to apply it to microarray data and write some more papers. So there are lots of papers, but I don't think there is a whole lot of new knowledge in them. Right? Okay. So the second term in this equation is a constant offset. Okay, wait, I think. Yeah. So the second term in this equation is a constant offset that simply shifts the RK versus GK scatter plot to a 45-degree diagonal line intersecting the origin. I think I probably skipped some pages maybe. Okay, so the second term in this equation is a constant offset that simply shifts the RK versus GK scatter plot to a 45-degree diagonal line intersecting the origin, and NQ is the number of probes that have measurement, it shouldn't be measurement quality score. This would be the probes where you expect that the ratio should be 1, okay? Because it's the same sample that is being hybridized both in the red and in the green channel, okay? So in some cases, the scatter plot may not be perfectly at a 45-degree diagonal line due to the difference when the scanner's two channels may operate at different linear characteristic regions. In this case, you're going to use full linear regression, right? Not just try to estimate the intercept and then shift it, right? So instead of requiring the line to intersect at the origin, may be necessary. And this is achieved by finding coefficients A and B to minimize this expectation, right? So expected value green minus a linear combination of the red, right? minimize that. Find AB to minimize this. So that's the other figure that I showed you. Now, some microarray expression levels may have large dynamic range that will cause systematic scanner deviations, such as nonlinear response at lower intensity range and saturation at higher intensity. Right? So all the data falling into these ranges, whenever you have saturation or you have nonlinear response, you will throw them out usually, right? The transition range without proper handling may still cause some significant error in differential expression gene detection, right? And to account for this deviation, some other guy came up with what he called LOES, or locally weighted linear regression, and that's regularly employed as a normalization method for such intensity dependent effects. There is a newer method that's called LOES. See, you change something a little bit, okay? Something you had like a beta distribution, you change that to a Rayleigh distribution, it's a new paper, okay? So I don't know whether it gives new benefit or not, but you have to sell it trying to say that it is going to give you new benefit. So there are lots and lots of papers like that, okay? And I haven't read most of them, and now they're 10 or 12 years old. If they were that important, they would now show up in books, right now. So the figure that I showed you earlier that shows the effect of these different normalizations, this is just the offset. That one is linear, right? Linear regression, and that one is lowest, and so on. And you could have more figures like this. Now, it may be that the two channels in a cDNA microarray represent two sources of messenger RNA to be compared, or that the red channel corresponds to the source of interest, while the green channel serves as a reference channel. In either case, a basic question is whether the red intensity is significantly greater than or less than the green intensity. And such a question is naturally approached in the framework of hypothesis tests, so you have to do statistical methods. Now, for a microarray having n genes with red and green fluorescent expression values labeled by R1, R2 up to Rn, and G1, G2 up to Gn respectively, the issue is whether Rk is over or underexpressed relative to Gk. Okay. That's what I've been talking about, right? And to determine that, you have to come up with the density function for the ratio. Yeah, go ahead. No, the n genes on the microarray, right? On the microarray, you have n spots, okay? Yeah, you can think of n, what reference? Oh, you mean probes, n probes. N genes on the microarray, okay? N complementary genes you have put on the microarray, right? So at each spot, the red intensity is a random variable, the green intensity is another random variable, right? So let mu sub rk, mu r sub k and sigma r sub k denote the mean and standard deviation of this random variable for the red intensity, right? And the same thing for the green intensity, g sub k, right? So the relevant hypothesis test is, the null hypothesis is that the red and the green have the same mean, right? That means there is no difference, right? And the alternative hypothesis is that the two have different means, right? And you can test this hypothesis using the ratio test statistic, which is the ratio of the red to the green, okay? So now there is biological plausibility, right? And that's discussed in that paper that I'm going to refer you to, right? So there is biological plausibility for assuming that the coefficient of variation is approximately constant across the microarray. The coefficient of variation is the ratio of the standard deviation to the mean, right? So in the paper that we are discussing, they provided some biological justification why you would assume that for each red sample, right, the ratio of the standard deviation to the mean is a constant, okay? That is constant across red and green, across all the spots on the microarray, right? So if you make this simplifying assumption of a constant coefficient of variation, meaning that, the sigma is c times the mean, right? Then under the null hypothesis, when the mean of the red is equal to the mean of the green, this equation will imply that the green and the red also have the same standard deviation, okay, under the null hypothesis, right? So in this case, you will be able to pool things together, right? Assuming R and G to be normally distributed, normally and identically distributed, the ratio, right, has the density function that's given like this, okay? I'm going to derive that, okay, later, right? Note the important thing is that the K does not appear on the right hand side, okay? Did you see that? For all the spots, okay, the density function is the same, okay? So owing to this constant coefficient of variation assumption, the subscript K does not appear on the right hand side of the equation. Hence the density function holds for all genes, and all ratios satisfying the null hypothesis can be pooled, right? So wherever red intensity is equal to the green intensity, right, you can take the samples from there, right, and do maximum likelihood estimation, you can use some other method too. You can go, this is the density function, right? So from this you can construct a maximum likelihood estimate for the unknown parameter C, and that is given in the on the next page, all right? So the estimate that you come up with is this one, okay? By taking the measuring the, so take all the spots, okay, where you assume that red and green will be of the same intensity. Maybe these are the duplicated genes, all right? Same genes you hybridize on different spots, okay? Or maybe from the same sample, okay? The red and the green, they are the same from from the same sample. Take that information and you can get an estimate for the C hat, okay? And so where these are the ratio samples taken from a family of housekeeping genes on a microarray. What are housekeeping genes? Anybody remember?
Yeah.
Go ahead.
The genes that are responsible for picking up the proteins and so on that are responsible for replication and these kind of things.
Yeah, they're there in every cell, you know. So ideally the housekeeping gene expression should not differ between the normal tissue and let's say the cancerous tissue, because both the cells have disease, the test sample and the the reference sample, okay, because the housekeeping, both of them have to do the housekeeping, all right. So on those you assume that the red and the green will have the same expression values, all right. The ratio should be close to 1. So you can use that information to calculate this parameter C hat. And this is the maximum likelihood estimate. I will walk you through the steps in the paper, right, next time, how to derive that density function, and how to also come up with this maximum likelihood estimate. Most probably I might just email that paper, the PDF file of the paper to you, right? And then on Monday in class I will go over that, And there is some more stuff here, but we can discuss that next time, That and a discussion of the paper, right? Let's stop here for today.